Thank you very much for coming to my seminar. I'm Bartek Waclaw from the University of Edinburgh in Scotland, and I'm a physicist, but I'm interested in microbiology, in particular in the evolution of drug resistance in microbes. And this is what my talk is going to be about. This slide presents a very brief overview what I'm actually going to discuss here. Imagine that you have a population of some microbes that expand in space, but the space is filled with an antimicrobial drug, so it can be an antibiotic of some sort. But the space is not uniformly filled with that drug. It creates a gradient, so the concentration is high at one end and low at the other end. This is actually where the population starts at this point. And what I'm going to investigate is how this scenario affects the rate of antibiotic resistance evolution in this bacterial population. And the result, which is maybe something that you could anticipate very easily, is that in the presence of gradients, what happens is that these bacteria, which are at the tip of an expanding wave of microbes, they will be a very strong selection imposed on mutants that are antibiotic resistant. And this is what you would see in the end, that susceptible bacteria stop at some point and resistant bacteria emerge at the tip and they create a new wave that is expanding in space. So this seems to be all very simple, but what I'm going to tell you in my seminar is, yes?
But that assumes that there is also a gradient of food, right?
No.
There's only
even if the food was distributed uniformly everywhere, but bacteria were confined, let's say, spatially, because they were growing in some sort of microfluidic device, they couldn't just grow everywhere, but only in one direction. That would still occur. So in reality, of course, you can think that in most situations, bacteria will grow in this direction simply because there's more food here, because they have already eaten all the food there, but it's not necessary for this mechanism to operate. So, so I'm going to explain you in a little bit more details what will happen when you have gradients of different strengths, and also how this compares to the uniform distribution of the drug, whether gradients actually speed up or slow down the evolution of resistance. And this is what my talk is going to be about. Okay, so before I really go into these details, maybe let me briefly tell you why this is all important. So 2 or 3 years ago, I would have probably had to explain you that we have this problem of antibiotic resistance rising in microbial populations, that more and more bacteria detected in human diseases are resistant to antibiotics. But now you all know this because it has become so popular and fancy topic that I don't think it's really necessary for me to convince you that this is an important problem. But of course, to tackle this problem from a medical point of view, which is eventually what we want to do, we first need to understand a couple of things, how resistance evolution occurs in microbial populations. And there are all these different factors that I think other people, not necessarily physicists, are better suited at addressing. But what I'm going to do is only to focus on the role of spatial structure and gradients and heterogeneities, environmental heterogeneities in populations. And I will show you some examples of real infections when I think such gradients are important and they can really affect the rate of evolution of antibiotic resistance. So maybe let me first discuss a simple experiment that was performed about 3 years ago in the group of Bob Austin, which clearly showed that antibiotic gradients can speed up the evolution of resistance, at least for certain antibiotics and at least for certain experimental scenarios. So in this experiment, what those people did was to construct a microfluidic device in which a gradient of the antibiotic ciprofloxacin was created across this device. And when they inoculated with bacteria, they observed that initially there were very few bacteria, so these are these bright dots here. These are the compartments of this microfluidic device which are occupied by bacteria. Initially, there was very few of them, but in a matter of 10 hours or so, bacteria spread to the whole microfluidic device, and they were thriving. Also, at this side of the device, where the concentration of ciprofloxacin was several hundred times larger than the minimal inhibitory concentration that inhibits growth of E. coli or of this bacterium. So this was yes.
Do you actually know why they use this strange geometry? I mean, why not something straight?
I think this is probably because you don't want to wash the bacteria out from your system. So this is why they've created these flows which are into opposing directions, and also they have this complicated network here because this somehow minimizes flows caused by the flow of the nutrients. So you have to pump the nutrient on both sides because otherwise the bacteria would not grow. You have to deliver them the nutrients, but you don't want to wash them away.
But effectively, the gradients go along the edges. I mean,
No, the gradient is created in the vertical direction. So this side has a lot of ciprofloxacin, this side has no ciprofloxacin, and the gradient is in this direction. So this was, I think, the first experiment that demonstrated that evolution of antibiotic resistance can occur within a couple of hours, which was kind of unexpected. And many biologists, I know this from Bob Austin, strongly objected to that notion that evolution can be that fast. So when I saw this and I spoke to Bob Austin, that was about 3 years ago, on the next day I had a model which would reproduce exactly what he saw experimentally. But then and also my colleagues, one of them is Philip Gurlich, who was sitting here in the audience, we started to think more deeply about this problem, and we thought, well, is it always the case that gradients speed up the evolution of resistance, or maybe it depends on the particular antibiotic or some geometrical properties of the environment? So we came up with a couple of questions that we wanted to know answers to. One of them was whether it is always the case that evolution speeds up in the presence of antibiotic gradients, such as this, or maybe sometimes you will have the antibiotic distributed uniformly. This will actually be faster. Evolution will be faster in this scenario. Then we also wanted to know what would happen if you have, contrary to what Bob Austin and his group observed in his experiment, in which each mutation increased the level of resistance, what would happen if we had a situation in which the population has to go through some sort of a fitness valley? So there is a mutation that you have to produce in the path, in the chain of mutations to the best, to the most resistant strain that has the resistance that is slightly lower than its predecessor and also the next mutant. And we also wanted to know how a repeatable biological evolution would be in the case of gradients compared to uniform environments. And by this, what I mean is that if we start from a susceptible bacteria and we have a resistant mutant which is separated from the susceptible one by a few mutations, does biological evolution always follow just one mutational path, one possible sequence of mutations towards this best fitted genotype, or maybe there are more pathways? So this is what we investigated in the last 3 years through several different models, which I'm going to explain very soon. But before I do that, I think it's necessary for me to tell you more about how we quantify the level of resistance. So this can be done in many different ways, but what we chose to do was to use a parameter that is called the minimal inhibitory concentration, which tells you how much antibiotic you need to stop the growth of a bacterial population. And this is a cartoon that shows how this minimal inhibitory concentration can be determined. So if I say I have a bacterial population that grows, it has some net growth rate, which is growth minus death. And this, in the absence of antibiotics, has some finite value. When I increase the concentration of the antibiotic, which what corresponds to the horizontal line in this plot, then this number will go down, and at some point it will cross 0, and this is where MIC is. And for the bacterium that is susceptible, the MIC will be somewhere here on that plot, and for a resistant mutant, the MIC will be somewhere further to the right. So it will be larger. And this plot here shows how such a growth curve looks for the specific antibiotic ciprofloxacin, and how you can determine MIC for ciprofloxacin, which turns out to be about 15 nanograms per ml. This is of course different for different antibiotics, and also the shape of this curve is not completely universal, but it always more or less looks like the sigmoidal curve. Okay, so now to the models and experiments, because I should have also mentioned that we have been studying this problem through computer models, analytical calculations, and also through some simple experiments. The simplest thing to do is to formulate an analytical model here, which is what my colleague Philip Grölich did, which assumes that the population of bacteria can be described as a set of individuals that reproduce or die stochastically with certain rates, and they can also migrate from one compartment over one-dimensional chain of compartments to another one, and each compartment has a different concentration of the antibiotic. I will explain you this in more details in the next slide. Then we did some computer simulations for the same model, but I also did recently computer simulations for more complicated geometries that resemble what is actually possible to happen in real bacterial infections. And lastly, I've been doing some simple experiments in which this one-dimensional model, which is possible to solve analytically to a certain extent, is basically experimentally reproduced in a microtiter plate in which I have a set of compartments, and I connect them, and I create a gradient by injecting different amounts of the antibiotic to different wells. I will come to that later, to this experiment. So let me now explain you more this very simple one-dimensional model, which was the model that we published in this paper in Physical Review Letters. The model assumes that we have a chain of connected compartments. Each compartment has a different concentration of the drug, and the concentration increases from the left to the right. And we also assume that bacteria that grow in these compartments, they can have several different types. So this blue bacterium is the wild type, which is the most susceptible type of bacterium to the antibiotic. But then this bacterium can also mutate and produce other genotypes that are more and more resistant to the antibiotic. So this is, of course, not very realistic from a biological point of view, because you never really have a one-dimensional chain of possible mutations, but I will come back to that later. So this is a more formal definition of the model. I'm not going to go through all these equations. If someone is interested, I can discuss it maybe later after the talk. But you can do it more formally, of course. And what you see then when you, say, simulate this model on a computer is a very interesting thing. Namely, if in the case of an antibiotic gradient, when the concentration of the antibiotic increases from left to the right here, if I run my computer simulation, what I see is that I have a sequential fixation of more and more resistant mutants in the environment, and this happens relatively quickly. Whereas if I do the same thing for a uniform distribution of the antibiotic, so now the antibiotic has the same concentration in each of these compartments, but the concentration is lower than the minimal inhibitor concentration for the wild type, so that bacteria can always grow in the system. If I do this, what I see is that now only the green mutant, which is the mutant with the lowest degree of resistance, evolves in this over the same period of time in the system. So here I have evolved a mutant that is hundreds of times more resistant to what happened in the uniform environment. And this is very interesting, of course. It shows basically what Bob Austin and his group has seen. But can it be explained or understood somehow more mathematically rather than by computer simulations? And the answer is yes. And one can formulate a very nice mathematical theory which allows you to calculate all the fixation times and so on. But I'm not going to do this. This is actually what Philip has done. I'm going to present here a much simpler theory which is more qualitative, but it also illustrates how this is all possible. So let's forget for a moment about gradients and special heterogeneities. Let's just focus on a single compartment of my system, which we can assume if it's small enough, it will be well mixed. There won't be any gradients there or heterogeneities. And let's say that there are n bacteria of type M. M is one of the mutants in this environment. What do I need to produce a mutant? Obviously, one of the bacteria must mutate. And this happens with certain probability. And then this bacteria must reach fixation in this environment. By fixation, I mean here that this new mutant, M+1, will grow and reach macroscopic concentration in the population. And these are the probabilities of these two events. So this first happens with a rate that depends on the mutation rate, the growth rate, and the size of the population. And then the fixation probability, as this is given by a standard formula from population genetics. This is basically the difference in the fitnesses of the two mutants. So when I now multiply these two numbers, it gives me the rate of emergence of new resistant mutants in my system. And if I now invert this, this will give me the average time to evolve a new resistant mutant. And this is given by this expression here. Now, how can I use it to explain my results for gradients and for uniform distributions? Let me first discuss the uniform distribution. So if I have a certain concentration of the drug in the system that is slightly below the MIC for the for the wild type, so that the wild type can grow, if the wild type produces a mutant which is slightly better adapted and can grow, it has a larger MIC, it will also mean that its growth rate at this specific concentration of the drug will be higher. And then the fitness difference between these two growth rates will be also relatively large if this point is close to the MIC of the wild type. But then if the green mutant wants to produce a new mutant, the red one, then this of course is supposed to be better adapted again, but now the difference between the growth rates of the green and the red one will be much smaller than previously. And you can imagine that if I wanted to produce the fourth mutant, then the difference would be even smaller. So every new mutant will have a slightly smaller fixation probability, and as a result of this, through this expression for the fixation time, the total time to reach a mutant that has accumulated several mutations and is very, very resistant to the drug will be very large. Therefore, in the case of a uniform distribution of the drug, evolution will be generally slow. All this assumes, and this is maybe something that I should make clear, all this assumes that I need several mutations to reach a significant level of resistance. Now, how does this work for the case of gradients? So if I have a gradient, then it means that my population will spread initially, but that at some point it will stop because it cannot go any further, because bacteria cannot grow at concentrations that are higher than their MIC. But then, if I produce a mutant somewhere here near the tip of my wild-type population, then the fitness difference between the mutant and the wild type will be large. So this new strain, this new mutant will quickly reach fixation. and it will spread a little bit further. And then the situation will happen again, exactly the same thing. Now the new mutant, the red one, which previously had a very small fitness advantage over the wild type, now will have again a very large fitness advantage, simply because the green one cannot grow further up at a higher concentration of the drug. And this is why now, if I calculate the time to fixation for each of the mutants, they all will be comparable. And the total time to reach the same level of resistance as in the uniform environment will be much smaller.
Can I ask a question?
Yes?
So, what's the difference in concentration? So, where's the average concentration of the uniform environment?
In the uniform environment, you set it to something. So this is a very interesting question, of course, because you somehow have to decide how to compare the two situations so that the comparison is fair. So what we want to do, we want to compare situations such that the time is minimal in each of the two situations, and the time is minimized in the uniform for the uniform drug distribution when C naught is very close to MIC1. So if so, this is how I determine the time in the uniform distribution. I set this C naught to be something very, very close, but slightly below MIC1. And then in the gradient case, what I do, I change the gradient either by fixing the total amount of the drug and just changing its distribution a little bit, or I even don't have to do that. I can just say that, let's say the concentration increases exponentially with a certain rate, and I change the rate, and I look for such a time that is minimal, that I can minimize with respect to some of the parameters that describe the distribution of the drug. And it turns out when I do this, this is what I think is a relatively fair comparison. The time to reach the best adapted mutant is in most cases much smaller in the gradient case than in the uniform case. So evolution of antibiotic resistance is generally much faster in the case of antibiotic gradient, simply because you have more space at the tip of the population. You don't have to compete with the previous strain, and therefore selection coefficients are very large. And if you need a couple of steps to adapt, this will take much less time in the gradient case. But this only works if gradients are not too steep, because if they are too steep, then what happens is the effective size of the population which is able to produce mutants, which is somehow given by the steepness of this profile here, and it's depicted by this black curve, might be too small. So there are two effects here. First of all, you have that effect that when you adapt many times for consecutive mutants, this will grow with the number of mutational steps necessary, and this will grow faster in the uniform case. But also another effect here is that the time to resistance will increase if the population gets smaller and smaller. And what is the size of the population that is relevant here is the number of organisms that are that they can reproduce at the tip of the population. So too steep gradients cause this effective population size to be too small, and then they effectively cause that evolution can be slower in the case of antibiotic gradients. So this picture illustrates something that I already told you about, which is that the more mutants you have, the higher is the difference between the times to best adapted mutant in the uniform and in the gradient case. If you have very few mutants, then it can be, say you need just 2 mutations to reach a certain level of resistance. Then gradients can be actually not so, they can be even slower than the uniform case because of that effect that comes from the smaller effective size of the population. But when you need more and more and more mutants to produce a desired level of resistance, then the situation reverses, and then what is really important is not the size of the population, but those fixation probabilities that get smaller and smaller and smaller with every step. So the more intermediate genotypes we have, the larger is the difference and the larger is the speed up in the presence of gradients. But this all is true only when we have a monotonous pathway to resistance. By this I mean the fact that each mutation increases the level of resistance. I can also imagine the following situation. Some of the mutations increase the level of resistance, but there is a mutation, and you cannot really avoid it, that temporarily decreases the level of resistance. And such scenarios are experimentally seen, for example, for streptomycin, if I correctly remember. So what must happen here is, since this mutant is somehow less fit, if the size of the population is not too large and the mutation rate is not too large, then what must happen here is that the green mutant must produce a double mutant that jumps over this fitness valley. And this will happen with a probability that can be very small if the mutation rate is small and the population is small, because this would happen with a probability that is mu square. And if this is a rate limiting step, which is exactly the case for n and mu being small, then what you see is that gradients can be slower than the uniform distribution because of that effect of a smaller effective population size. Now, this population that is at the tip of my expanding wave of bacteria, it has to produce a double mutant. And this rate will be proportional to mu squared times the total size of this population at the tip. And in the uniform case, the effective population size is much larger because this is basically the total size of the population in the system. Of course, there is this complication because you have to multiply both probabilities by the fixation probability of a beneficial mutant. But it turns out that in certain scenarios, this number is comparable in both cases. And then what happens is gradients slow down the resistance evolution. So what this plot shows here is I plot the time to reach resistance, by which I mean 5 mutations, 5 separate mutations, versus the concentration of the drug in the system. 1 is, it corresponds to the MIC of the wild type. So above 1, nothing can really grow. Below 1, I always have some growth. And this, in the uniform case, doesn't seem to depend very much on the concentration. The time to resistance is more or less constant. Whereas in the case of antibiotic gradients, it turns out that the steeper the gradient is, the higher is the time to resistance, because I effectively reduce the size of the population by squeezing it through this gradient only to one side of my compartment. Okay.
These are simulations.
This is from, the points are from simulations, and these curves are analytical calculations. Last thing that I wanted to say about this theoretical approach is what happens if I have more sophisticated, more realistic pathways to resistance. So as I already said, a one-dimensional pathway in which I have a chain of mutations, each of them increasing resistance, or perhaps some of them can produce a fitness valley. This isn't very realistic biologically. What is more realistic biologically is that is very well illustrated by this example of a sort of a resistance landscape, which was mapped by Daniel Weinreich and others in this paper in Science from 2006, in which they measured the MIC for all 32 mutants of a gene TEM1, which is a gene coding for a protein called beta-lactamase that degrades cifotaxin, which is the antibiotic that they used. They introduced all possible combinations of 5 specific point mutations. If all mutations were present, then the resistance was the MIC was 4,100 nanograms per mil, whereas the resistance of the wild type was only 0.09 nanograms. No, sorry, this is in micrograms per mil, and this is also in micrograms per mil. So the difference between the two numbers is by a factor of 10 to the fifth, more or less. And they estimated the MICs of all intermediate mutants. And what they saw was that if they assumed that they've never actually done this experiment, but when they assumed that the evolution would, when they did computer simulations of a situation in which they assumed a well-mixed population of bacteria evolving in this sort of resistance landscape, they saw that only very few pathways, which are these green, two green pathways, were selected by biological evolution. So what I wanted to do here, I wanted to replicate the same thing in the case of antibiotic gradients. So first of all, what I saw, and I more or less repeated what they did, was that, okay, there was only very few pathways that were selected by biological evolution, from the wild type to the best adapted mutant. But there was also a considerable variation here. So it wasn't just one or two pathways. There were pathways which had a little bit smaller probabilities, but they were nevertheless, they sometimes occurred in the simulation. When I did this in the gradient case, I saw, initially to my surprise, but later I convinced myself that it is actually quite obvious, that only one pathway, plus a little bit more, but with much smaller probabilities, is selected. And so what this picture shows is that gradients can actually cause evolution to be more predictable, more repeatable when you do the same experiment over and over again. And I think this is very interesting. And the reason why I think it is so is actually very simple. So in this case, if you remember, if the distribution of the drug is uniform, then selection coefficients for consecutive mutants are getting smaller and smaller and smaller. So at some point, evolution essentially becomes neutral. And when you have neutral evolution, then there is no reason why certain pathways should be preferred over other pathways. Whereas in the case of a ciprofloxacin gradient, there will be always selection. There won't be any neutrality there. And then the bigger the selection coefficient is, the higher is the chance that this pathway will be selected. And the pathway that is actually selected here is the one with a larger selection coefficients. So that's, I think, the simplest explanation for that. So this is all I wanted to say about computer simulations of the simple model. Do you have any questions to this part? Yes?
Just a completely different question. So this is for adaptation to drugs or antibiotics. Yes. Could you also use a similar approach to look to adaptation to resource utilization, for instance?
Yes. So the problem of adaptation to non-uniform environments, this is something that many population geneticists studied in the past. And we, in the sense, are not very original here by saying that gradients can speed up the process of evolution, because in the sense, this has been already known, but not specifically for antibiotics. So what we have done here is to somehow adapt this all knowledge from population genetics to the problem of antibiotic resistance evolution. So, you know, when you looked at the literature, you will never find statements that are exactly like our statements, but it's something, you know, many people have come very close to making statements such as this.
But the point is that this can be inverted and sometimes it slows down.
Yes, yes.
So yes. This is, I think, a point that for short mutational pathways, it slows down, or when you have fitness values, it slows down, which is
yes.
somehow
yeah, but what I just wanted to say was that, you know, in literature there are many examples of situations in which you have faster evolution when you have environmental heterogeneities. So that is something that is maybe not completely novel here. But these other things about pathways to resistance, or like Philip just said about what happens in the presence of a fitness valley, that's something that I haven't really seen before. Okay.
Can I ask another
Yes.
generic question? When you've spoken about the genome and mutations of
Yes.
mutations in the genome, but what about other sources of resistance, which say protein levels, which would be inherited only, say, for a couple of generations?
Yes, that's a very good question. That's a very good question. And this model doesn't really make any difference between genetic and phenotypic resistance. You could also think about some of these mutations as phenotypic switching between different states of the same cell. So the cell has the same genetic material, but it just switches between different states. And of course, the probability of such a transition would be typically larger than the probability of a mutation. One could imagine doing the same experiment with some of the transitions being much more likely than other transitions. We haven't actually done anything like this, but I thought about it, and I'm almost 100% sure that things like this happen experimentally. And this is what I wanted to come to now. So all this modeling is very interesting, of course, but the ultimate question is here whether it agrees with some biological experiments. And the answer is very complex, and I'm not completely sure yet. But I'm going to discuss just a couple of findings, experimental findings that I've done in this experimental setup. So as I already mentioned before, the way how I do these experiments with gradients is I take a microtiter plate and I cut holes between wells in a single row of wells in the microtiter plate, and this creates a chain of connected habitats. Then I fill them with some medium. I add different amounts of antibiotics to each of the wells, and in that way I create gradients that go either from right to the left or from the left to the right. And I inoculate this set of connected compartments with bacteria on the side where there is very little antibiotic. Then I put this into this strangely looking machine, which measures the optical density in each of the wells every hour or so, and the optical density here correlates with the size of the population in each well. So by doing this, I can tell where the bacteria are at a given time, how far they have spread in the environment against the gradient. And the nice thing about this experiment is that since this is done in standard microtiter plates, each such plate can have up to 16 independent experiments, and then I can also use this machine called a stacker to stack multiple plates and to read them sequentially, so that in the end I can run hundreds of experiments simultaneously, which gives me quite a good statistics, which is very nice for a physicist. So it's not just 3 replicas, but hundreds of replicas. Okay, and these are some videos from the experiment. What is shown here is the green color shows the optical density, which is more or less proportional to the size of the population in each well. And this is a series of time lapse, which shows how the population spreads in the environment, starting from no antibiotic and goes against the gradient. And it stops somewhere here. So this experiment has been done over the period of about 4 days. So it has never reached the highest concentration. It was 50 times the MIC. But the concentration that it has reached was about 20 times MIC. So it has evolved resistance to ciprofloxacin by a factor of 20.
How do you keep the gradients constant?
Well, so I get this question very often when I present this talk. This was a key challenge in this experiment, and the idea that I came up with was to create very tiny holes between these micro compartments, so that there is very little diffusion. That's one thing, but another, and this is even more important, is to stabilize the medium so that it doesn't flow around when the microplate reader moves the plate to read the density, the optical density in each of the wells. So what I do, I add a little bit of agarose to each well. And then when I cool it down, so I do it at 60 degrees, then I cool it down, and then it solidifies. It creates a very soft gel. Well, it's not completely solid, but it's a very soft gel in which the gradient turns out to be pretty stable. There is, of course, a little bit of diffusion. Sorry?
Do you have any plot to show this?
Not here, but I can show you on my laptop later. I don't have it in the presentation, but the gradient it's not absolutely stable. Of course, there is some diffusion, but it's over this period of time, it doesn't change significantly. There is another problem here, which is drug degradation, but for ciprofloxacin, this turns out to be not a very big problem because ciprofloxacin degrades very, very slowly in LV. So, so you see that at least some of the experiments here, in some of the experiments, there must have been some evolution because bacteria managed to grow to be able to grow at a concentration that's 20 times their MIC. The question is whether this is genetic or whether this is phenotypic, and I will come to that.
So in your bottom gradient, where would 20 times MIC be?
I think it would be, so this is all exponential, so I would say somewhere here. No, sorry, no. No, I don't know. No, it must be somewhere close. It must be somewhere here, I think. So, so what it shows is for steeper gradients, it seems to be very little evolution, whereas for gradients that are more shallow, there is some evolution, but not in each of these experiments. It means that this is a stochastic process. It sometimes happens, sometimes it doesn't. This is in qualitative agreement with the model, but to get a quantitative agreement, this is something that I still have to work on. Okay.
Excuse me, sorry, about that. How do you measure the concentration of antibiotic in a given place?
I don't measure it, so I add a prescribed amount.
How do you know that the gradient is stable?
Because I did an experiment with a dye in lieu of the antibiotic. I used.
And you assume that the dye is the same as antibiotic?
Yeah, it has the same. Well, it's not exactly the same. Of course, one could argue that the molecule is not exactly the same molecule, but it has similar molecular weight, which means that the diffusion coefficient should be very similar. And this is how I know that the gradient is more or less stable. And I also made computer simulations which show that the gradient should be stable.
So the mechanism of resistance in the antibiotic, that doesn't metabolize the antibiotic when it's, as the organism gets better at resistance?
No, sorry. So what happens here once the bacteria started to grow in a given well, that is something I don't know. But the gradient is stable on that side. So as long as bacteria have reached a certain well, then of course there will be some metabolism, and then I cannot really tell anything about the concentration anymore. But the hope is that you have to initially have just very few bacteria in that well to be able to reproduce, and they cannot really metabolize that much of the antibiotic. So this is why I think there is still some antibiotic at the tip of the moving front of the population. So let me now briefly discuss some genetics of this process. So I've done some DNA sequencing for about 28 samples taken from my experiment. This picture shows 4 samples. And these diagrams, they are sort of chemographs. So what is plotted here is the position of the population. So this is this boundary here as it moves in time. And what you see is, for example, in sample 1, population initially expanded at some velocity, which was more or less constant, but then something happened and it began to expand at a smaller velocity. this line here, the slope of the line determines the velocity of expansion basically in those plots. But in other samples, like in this one, the situation looks a little bit different. So this already might indicate that there has been a considerable variation in how bacteria evolved resistance to this antibiotic in all those experiments. And this is indeed what I showed when I sequenced them. This picture here shows all genes that have been mutated in those 4 experiments, and you see that, for example, in the first experiment, this gene, which is a component of gyrase, DNA gyrase, has been mutated, but in this experiment it has not been mutated. And so there is considerable, yes?
Just one thing to the previous, like the diffusion. What is the diffusion constant? The thermal diffusion constant of the dye or antibiotic when you just take?
It's on the order of 10 to the minus 6 centimeters square per second, if I correctly remember.
Per second.
Yeah. So I think once I calculated it, it would need several weeks for the antibiotic to really diffuse in the system.
Yeah.
Yes.
Okay, and you look only for.
For 4 days. So there is some diffusion, but the profile is not infinitely steep.
But it's probably less than 1 chamber within.
It gets smoother and smoother over time, but it's not very significant. The change is not very significant. And anyway, you should remember that all these results are qualitative rather than quantitative. Even if the gradient changes a little bit, then this is not expected to change qualitatively the results. So anyway, so I found in these experiments that different genes have mutated in different experimental samples, but what is somehow what suggests that this sequencing has been done correctly is that all these genes have something to do with antibiotic resistance. They have been reported previously as being responsible for increased levels of resistance.
Sorry, so you just sequenced at the end of the experiment?
In this slide, yes. But I've also did some sort of fossil track record for 3 different populations. I'm going to do more of it because this was just a pilot experiment. I just wanted to see how it would work. But nevertheless, I did it for 3 different experiments. And I sequenced the population at 4 different spatial locations. And what I saw was something very interesting, namely when I order all these genes, the way how they are aligned in space, which also corresponds to the way how they are the concentration of the antibiotic changes in the same direction as the spatial coordinate changes, and this also corresponds to time here. So this first sample corresponds to the smallest concentration of the antibiotic, and this one corresponds to the higher concentration of the antibiotic. What I saw was that initially there was a considerable variation in the genes that were mutated, but then it went down and down, and in the end, in this experiment, this was at the highest concentration. I saw only very few genes mutated. So these must be the genes that really give the fitness advantage to the cell, and the rest has been just so these genes here that you can see, they must have been present in a small fraction of the population. This is why I detected them. But what really spread to the higher and higher concentration of the antibiotic was only those cells that had those 6 or so genes mutated. And a very similar situation happened in the second experiment, but this should be the third. And in the third experiment, it was slightly different. I saw very little change as I moved along the gradient. It was the same experiment. So what it shows is that there is not only variability between what genes are selected, which means that probably different combination of genes gives you the same fitness advantage, but also there is large variability between how variable the population, how genetically variable the population is. Because the study experiment, these were all identical experimental conditions. Same thing repeated.
But there isn't identical life, so could you tell us a little bit more about how your experiments are done?
Yes, if you want, but it means that I won't be able to tell you more about
So
clinical
maybe-
scenarios.
But you inoculated with the same clone?
Yes.
At
Well,
the-
sorry. Well,
Or the
I.
clone from the
It should be. It should be the
same
same.
growth?
It should be the same clone. It is always. I use always the same frozen stock, which should be monoclonal. But in reality, who knows?
And how many cells grow in such wells?
I think that would be on the order of 10 to the maximally 10 to the 9th, but I think it's between 10 to the 9th.
You didn't mention, but it's E. coli.
Yes, it's E. coli. Yes,
What
yes.
strain?
It's MG1655. Yes.
So the color, right, it tells us the frequency of mutation in the population.
Yes.
So does that mean in the last experiment, in the last time point, you had some that were not mutated at all?
Yes, which seems strange, but I think it was because my sample got contaminated. Because I have these results here, they come from sequencing of mixtures of bacteria from each of those wells. But I also did I also sequenced some isolates from the wells, and for this experiment I had an isolate from the same well, and when I sequenced it, it had all those genes marked red, so it was 100%. So it must have, this sample must have gotten contaminated somehow.
So what's then the frequency, the coverage of reads on a particular locus?
Yeah, it means how many reads have this specific. No, sorry. In what proportion of the population has this specific gene mutated? Because in these different experiments, it often occurs that the same gene is mutated in different places. So what I don't really look at, what specifically happened there, but I'm just interested in whether a gene has mutated or not, and what was the proportion of the gene in the population, the mutated gene.
Someone else would suggest that it looks like that the evolution is highly non-neutral concerning all the other genes, because it seems that, in the beginning it seems to be neutral, there are many mutations that somehow pop up, as you would expect from neutral evolution. But then somehow they're all eliminated, so it becomes highly non-neutral. Also those that are not directly involved into resistance become non-neutral, so they, like as if they were deleterious.
It might be, yes.
Yeah.
Well, definitely there is selection here. This is how the experiment is
Okay,
concerned.
there's a selection for a certain part of the genome, of course, but that's like, if you take the whole genome, and it seems like to affect also like the things where you would expect it.
Yeah. So you will see mutations that correspond to genes that give you a fitness advantage, but you also see passenger mutations. So in some cases, some of these genes, this one, for example,
like eliminated. The passengers seem to be eliminated.
some of them, but probably not all. So there is many genes here, like this one, for example, which has never been reported to have anything to do with antibiotic resistance. Maybe this is just a passenger mutation. Who knows?
Yeah, but right, yes.
Yes.
I have two questions now. Is this complete random shotgun sequencing of the whole genome, or targeted to?
This is whole genome resequencing.
Okay. And the mutations that you're reporting, do they include silent mutations? So anything that deviates from the wild type sequence?
Yes, I think yes. Because no, wait a second, I have to think about it. I had two data sets, and this one I think this one it includes silent mutations. But I think for many of those genes that are listed here and they seem to be changed, I explicitly checked that, and there were no silent mutations. Some of them are probably silent, but not many.
So, for example, at the beginning where you have sort of your blue genes where you've got low frequency,
It can be
some
that
of those
some of them will be
and
silent.
obviously they're not going to have any
Yes,
effect on
yes,
the
yes,
phenotype, so
yes,
then they won't
yes,
persist.
Yeah, some of them could be deleterious, like Philip mentioned, and they would be also deleted. Okay, so my time is running out, and I wanted to tell you also something about the significance of all of this to real bacterial infections, but I don't really think I have that much time. So maybe I will just briefly skip this and tell you about 2 examples very, very briefly. So the question is, do gradients occur in vivo or not? That's the problem that I want to know the answer to. And one, of course, cannot be certain because nobody has done such experiments in vivo, but I've done numerical simulations that show that it is plausible that gradients occur in vivo in certain situations. What is required for that is that you have an isolated area in the tissue or somewhere in the body when there is not much blood flow, because when there is a lot of blood vessels nearby, then it turns out that any drug will quickly diffuse into the tissue and there won't be any gradients. But in a situation such as this, in the lungs, when you have these bronchiolae, which in patients that suffer from cystic fibrosis can be filled with mucus, and this mucus often becomes infected by Pseudomonas aeruginosa, there is enough distance from blood vessels. So inside the mucus, this is a picture from a simulation that shows a section to some of these bronchioli. Inside here, there will be possible for the bacteria to evolve resistance because they have the time it takes for the antibiotics to diffuse into these bronchioli will be relatively long because they are large enough. And this simulation shows this. So the population that is initially susceptible dies out when I treat it with antibiotics. But at some point, what I see is the emergence of these resistant drugs, resistant mutations. And there is some evidence that shows that indeed resistance can evolve in vivo in those patients that suffer from cystic fibrosis. So it is at least plausible that this is because of such spatial temporal gradients. And another example that I wanted to briefly mention is infected wounds, which often become necrotic, which means that there is not enough blood flowing into the wound, and then antibiotics cannot really penetrate that wound, and what you have to do then is to apply antibiotics topically on the wound, on the surface of the wound. But when you do this, this is what is shown in the second animation here. The antibiotic has to diffuse from the surface into the wound. This is an expanding bacterial population. Now the antibiotic starts to diffuse into the wound, and at some point it starts killing the bacteria, but because of the gradient that it creates, it also promotes the evolution of resistance, which is shown here. And in this situation, you cannot really eradicate the population of bacteria. So that will be the end of my talk. And just to briefly summarize it, what is most important, I think, in all of this is that gradients often, but not always, can, as we have seen, can speed up the process of evolution of resistance. And also they can make it more repeatable, although this is maybe less, this is kind of obvious in simulations, but less obvious from experimental data at the moment. And gradients can also promote de novo evolution of resistance, at least in some diseases, provided that there is enough time and space for gradients to occur. So thank you very much for your attention. Do you have any more questions? Yes.
Like the sequencing. When I think we discussed about that before, when you have like many things that somehow you connect, you know already how you can connect the mutations in some kind of pathway, of mutational pathway. Do you have already an idea how this can be done, how you can connect it?
No. But this is something maybe we should discuss later, because
Yeah,
I don't
okay.
think it's interesting to other people.
was wondering if
Yes.
Did you try the sequencing when you didn't have a gradient?
No. So this was just a pilot experiment. So I didn't do it for the other case without any gradient. This is something I want to do definitely.